Hi guys, this is Biology Buddies and I am Chloe, the presenter. And today I am going to talk about the meiosis process. The process purposefully for producing gametes in sexual reproductions. Also keep in mind that the meiosis is the nuclear divisions that result in the genetically identical cells for haploid cells. So, unlike the mitosis, which produce the diploid cells, the meiosis produce the haploid cells, which is half of the chromosomes, half the amount of chromosomes that the diploid cells consist of. Right? So, now, let's talk about the importance of the meiosis. So, the meiosis is very important and crucially important for the sexual life cycle as it allows the fusions of gametes, or we could call their fertilizations to happen. And as we know, the fertilizations doubles the number of chromosomes. Hence, it is very important that the chromosomes number is also halved. Otherwise, the chromosome number would always keep doubling every generations. And this is not ideal. This is not ideal at all, right? So, just keep in mind that having the having of the chromosomes occurs during meiosis, and in animals, this havings occurs during the creations of gametes. And so, to be specific, it happens in animals. It happens in the testes of male animals and in the ovaries of their female animals. So the gametes are the sperm cells and the ova. On the other hand, when it comes to flowering plants, their meiosis occurs in the antlers and the ovaries, and their male gamete is carried in the pollen of their plants, the flowering plants, and their female gametes are carried in the ovules of their flowering plants. Moving on, let's have a general look at the process of meiosis. So here you guys could see, first of all, there will be interphase occur and prior to the meiosis, and so their meiosis process involve their meiosis 1 and meiosis 2. So here is some more specific pictures of their meiosis 1 and 2. So in meiosis 1, it is called the reductions divisions, and the number of chromosomes per cell is reduced by a factor of 2. And under meiosis 2, it is obvious that there is no interphase, and the interphase only occurred in the meiosis 1. So, you guys still remember this? Still remember the interphase that I've already mentioned in the mitosis video? If you do, congratulations! I'm really proud of you. And if you haven't, it's okay though. And so if you haven't checked it out yet, then why not go to our mitosis video to have a discover or revisions after mitosis, at the same time understand the interphase this phase better, right? So, yeah. Let's now move on to their main stage, their meiosis stage, specifically their meiosis 1. So in their meiosis 1, start with the prophase 1. So for the prophase 1, these are what will happen. And so their centrioles will migrate to the opposite poles. Then their spindle fibers will be formed from the centrioles, or we could say the centrosomes. Then the nuclear membrane will dissolve. Following that is the condense of chromosomes, as the chromosomes will be shortened, become denser, and become more visible. Then their synapses will occur as two homologous chromosomes with the exact copies of each other and have their exact same length except for the alleles difference. That two homologous chromosomes will align alongside each other, and when the homologous chromosomes pair up in a close proximity, that two homologous chromosomes will get entangled and their crossing over will occur. Now I will talk a bit about the crossing over. So the crossing over is when the homologous pairs of chromosomes connect via the chiasmata between the non-sister chromatids of their two homologous chromosomes, and when they connect via the chiasmata like that, they will form bivalents. And so the chiasma or the chiasmata, which is the plural noun of the chiasma, is the point where non-sister chromatids exchange alleles by the break of a DNA sections from one chromosomes to rejoin with another chromosomes. So that kind of breaking and rejoining of DNA sections at their chiasma is called the recombinations, and the recombinations are catalyzed by endonuclease and DNA ligase. Then the recombinations will result in recombinants, and the recombinants are just the chromosomes that have already exchanged their DNA molecules. So accordingly, the recombinations enhance the creations of new combinations of genetic informations. Also, keep in mind that there are infinite combinations of recombinants as a result of recombinations. And thanks a lot to crossing over and recombinations, because those processed, the crossing over and recombinations really help the species to ensure the genetic variations and ensure that species can adapt and evolve over time. Following the prophase 1 is the metaphase 1. So in this phase, the bivalents will be lined up at their equator by spindle fibers, as their spindle fibers will attach to the centromere of the bivalents and line up the bivalents via random assortment. So let's talk more about the random assortment. So random assortment will produce different combos of alleles due to the random alignments of homologous pairs or bivalents along equator. So when homologous chromosomes line up, each pair can be arranged with either chromosomes on the top. So the number of different possible chromosome combinations or gamete combinations are 2 to the power of n gametes combinations. And so the n here is the number of chromosomes in a haploid cell, which is half of the diploid cell. So in humans, is comprised of the haploid of 23 chromosomes. So humans will have 2 to the power of 23 gamete combinations via random assortment. Now let's move on to a less complicated phase. So the anaphase 1, which follow the metaphase 1. So in the anaphase 1, the spindle fibers will contract and pull the whole chromosomes to opposite ends of the pole. So the homologous pairs of chromosomes will be separated here. However, the centromeres do not split. Finally, to the last steps of meiosis 1, the telophase 1 and the cytokinesis. So in here, the chromosomes, once arrive at the poles, they will decondense, and their nuclear membrane might reform, and their cytokinesis will start here as cell divides, forming 2 haploid daughter cells. And prior to the meiosis 2, there will be no interphase, but there might be interkinesis. And so in the interkinesis phase, their microtubules will reassemble into 2 new spindles for second divisions. And in this phase, there will be no further replications of chromosomes. And just a little fun fact, this process may last for years. Head to the phase 2. In this phase, the nuclear membrane will dissolve again, and the centrosomes will move to the opposite poles, and spindle fibers form a right angle to the old one in meiosis 1. You guys should remember this because it's very important. Then the chromosomes will condense, and the same go for this daughter cell. After that, the metaphase 2 will occur, and in this phase, the spindle fibers of the centrosomes will attach to the centromeres of chromosomes and align them along the equator. So yeah, same for both daughter cells. As the process get onto the anaphase 2, the spindle's fibers will contract here and separate their sister chromatids at the same time. Their centromere will be divided, and their spindle fibers will move the chromatids to the opposite poles, and the separated chromatids are now no longer sister chromatids, but instead they are called single chromosomes. And yeah, the same go for both cells, like both daughter cells. Final stage is telophase 2 and cytokinesis. So their chromosomes here in this phase will decondense and the nuclear membrane will reformed and their cytokinesis start to happen as cell divide to form 4 haploid daughter cells. So yeah, that is all about their meiosis, meiosis 1 and meiosis 2. Oh, and now I want to talk about when their meiosis doesn't go right. So when their meiosis doesn't go right, such as non-disjunctions, this will cause genetic diseases such as Down syndromes. So there should be something to control it. And thanks to the cyclins, there are proteins that control cell divisions like meiosis and mitosis process. So yeah, if you want to know more about how cyclins work, please check out our video cyclins for more information. And thank you for now, and hope to see you guys again soon. Bye bye.